So lab test 2 for the review. Let's move ahead. And we're going to start with exercise 5 on enzymes. Page 57 is where this starts. Page 57. So what I did is I just put a chemical reaction up here. And you know, I can tell, this may bother some of you because it's not balanced. Okay, good. Will. Doesn't bother Will. But anyway, we have H2O2 in the presence of catalase forms water and oxygen. And for number 1, we're supposed to identify the reactants. So what would be the reactants of the chemical reaction I have on the screen right now? H2O2. So I'm just going to box that and put an R. Okay, and then the next thing was products. What are the products for this reaction? Water and the oxygen. So here's our products.
And the plus sign.
Yep. Okay, and then I want to add something. I'd also, for us to also be able to identify the enzyme of this chemical reaction, what would be the enzyme? Catalase. Very good. So I want you to get used to seeing it this way, where reactants, of course, are on the left, products are on the other side of the arrow on the right, and then the enzyme is written above the arrow. How else can you tell that this is an enzyme? ASE. James is correct. The ACE tells you it's an enzyme. Okay, everybody okay on number 1? Being able to do that? Super. Okay, number 2. Now, we could go through and define each of these words, but would be more applicable to our test is to look at page 57. On page 57, we have a chemical, a piece of artwork at the bottom of page 57. It kind of looks like this. It's not exactly like this, but we could pretend that it looked like this, and we would have all of the words erased away. See how I erased the words so you couldn't see them?
That's not what an erase is.
Well, I scratched them out. Thank you, sir. And so I would need you to be able to tell which one was the enzyme and which one was the substrate. Where's the enzyme substrate complex? Where's the product? Hi.
I'm Jake Murphy. I emailed you last semester.
I remember now. You want to come up and get a review sheet? And is that going to be comfortable for you? Remind me your first name again?
Jake.
Jake. Got it. Glad you could join us, Jake. Okay, so on page 57 in our lab manual, then you would have to pick out what is the enzyme substrate active site, enzyme substrate complex, and what's the products. Now, I've had students only have trouble with one aspect of this, and that is figuring out which one's the enzyme and which one's the substrate. Okay, and so I just want to comment real quickly. Most students tell me they can spot the enzyme because of these indentions in the shape of the molecule. What are those indentions called? The active site. And that's fine. That would be a good way of knowing. Here's how I do it. I can tell that this is the enzyme and the top one is the substrate because the substrate changes. Right, check it out. The substrate changes in the artwork. What's the substrate change into? Product. Does the enzyme change? No. So in case while you're looking at the artwork, if you think, oh, I'm not sure, look who changes. Whoever's changing is the substrate. Whoever's staying the same is the enzyme. And then once you decide, you should be able to look at the enzyme and figure out where the active site is. You're okay on the artwork. And again, page 57 is a great place to think about the same idea about the artwork. Okay, I also like the second part of number 2. Everybody look at the second part of number 2. What's the effect of an enzyme on the activation energy of a chemical reaction? And so I drew a graph up here. I don't know if you can tell. It's kind of sloppy. Oh well. First of all, this chemical reaction is either exergonic or endergonic. Can you tell me what it is? This one's exergonic. Good job. Exergonic because the reactants have more energy than the products. Now, it's got 2 different colors on it. This one's kind of a yellowish green. This one's more of a red. Got two different lines on there. One of these lines would be the line that would represent using an enzyme. Which one is the line representing an enzyme is used?
Yellow.
The yellow one. And how did you know?
Because of the energy.
The energy of activation. Everybody look at the energy of activation, which is the amount of energy that has to be put into the chemical reaction for it to occur. The energy of activation is lower if you're using an enzyme. Okay, and you have to know that. If you didn't know that, you have to know that. If you use an enzyme, it lowers the energy of activation. Lowers it. But okay, on number 2. Okay, number 3. What happens when an enzyme is denatured? Tell me what happens. It dies. That's not it.
It loses its abilities.
It loses its shape.
Loses its shape. And then Grant added, it loses its ability to work. Yeah. So it stops working. So is that good or bad? That's bad. If your enzymes stop working, what could happen to your cells? They die. That's right. Okay, second part of number 3. What conditions might cause this to happen? Tell me again. Is that Myra? Tell me again.
And
pH and what else?
heat.
That's very good.
Like the basketball.
I like the heat, yeah. So I'm going to go with high temperature, which was what she said was heat, and then extreme pHs, like really acidic or really basic. Could be really high on the pH scale, which would be basic. Could be really low on the pH scale, which is acidic. And that's true for most enzymes. Okay, on number 3. Okay, number 4, what's the function of catalase? Well, if you don't recall, it's on page 58, everybody, page 58. How would you describe what catalase does? Excellent. Speeds. Hey, Kat. Speeds the breakdown of what? Hydrogen peroxide, which is H2O2. Second part of number 4. How did we judge? That means how did we know if the reaction was occurring? How did we know? Looking for the bubbles. What was the identity of the gas in the bubbles? Thank you. Oxygen bubbles. We looked for oxygen bubbles. Oxygen bubbles. Okay, number 5. What effects do different temperatures have on catalase activity? Now, I have that you should have data on page 60 for this. Let's all turn to page 60 to glance at our data. Okay, did catalase work really well or not very well when it was cold?
Really well.
Really well. So I'm going to give it a big high. Really good. How about warm?
Not so much.
Yeah, not as much. Okay, but it was good. How about hot?
No.
No, it was bad. It was bad. Okay, so why did catalase work so poorly in the hot?
It was denatured.
It was denatured. The catalase was denatured. That's good. Okay, that was too big. Back to 13. Okay, I just want to just remind you that catalase that we used was from a potato. So I wasn't surprised that it worked well for cold or for warm because potatoes live in the ground. And you know the ground can get pretty cold, right? It also can get pretty warm. So if you had good bubbling for both of those, that is to be expected. One last comment on that. Catalase is a universal enzyme, which means it's found in all kingdoms and domains of life. It is found in all living cells. The ability to break down hydrogen peroxide happens in every cell. Okay, number 7, what's the effect of a pH outside the optimum range of an enzyme? If you go outside, let's say 6 to 8 on the pH scale, what happens to the enzyme? Like if you have a pH of 4 or a pH of 10, what do you think the enzyme would do? It would denature. It would not work. Yes. Oh, did I skip one? Yes, I did. It's because we're omitting number 6. Thanks for commenting on that. We did not do that activity, so we are omitting number 6. Thank you for pointing that out. Yeah, so omit number 6. On number 7, what's the effect of a pH outside the optimum range of an enzyme? The enzyme will denature. What would HCl and NaOH do to catalase? Well, HCl is an acid. NaOH is a base. So they're going to denature catalase. Make it stop working. Now, class, I just want to make sure that you agree with me on that. Page 62. This goes with number 7, page 62. I think that's what we did, is we put catalase in test tube number 1 with some acid. Is that right? And then we put catalase with some sodium hydroxide in number 3. So I drew those 3 test tubes. Check it out. I have them 1, 2, 3. In number 1, we had HCl. Number 2, we just had distilled water. And number 3, we had NaOH. And so we put in catalase, then we put in the acid, the water, and the base. Then we had to wait a minute, and then we took the pH, and then I think we added the hydrogen peroxide. So I want to ask you, from your own data, what happened in test tube 1? Did you get bubbles? No. So no. Test tube number 2, did you get bubbles? Yes. Test tube number 3, did you get bubbles? No. Now, I'd like to talk about your pHs, please. Test tube number 1, what did you get for the pH? A 1? Does everybody agree on that? Okay, test tube number 2, what did you get for the pH? 6. Anybody get 7? But we all agree on 6? Okay, and test tube number 3, what did you get for the pH? 14. Wowee. Okay, so it looked like catalase worked best right here in the middle, which is near neutral. We weren't exactly at neutral, we were near neutral. This is a really low pH and it wouldn't bubble. This is a really high pH and it wouldn't bubble. So earlier Myra told us that really low pHs or really high pHs will do what to an enzyme? Denature. And our data supports that. Just want to point out your data does support that conclusion. But okay, on question 7. Okay, let's glance back at the exercise on enzymes. Remember, we're omitting number 6. Is there anybody who needs anything repeated about enzymes? This previous one? That one right there? You bet. Anybody else about enzymes? Okay. All right, well, we're going to roll up our sleeves now because we're heading into photosynthesis and cellular respiration. So the next thing we're going to talk about is exercise 6, photosynthesis, which I believe is on page 67. So would turn to page 67 in your manual. So help me out. What are the reactants? See if you can do it without looking in your book. What are the reactants of photosynthesis? Go ahead.
CO2 plus H2O with solar energy.
I'm going to put a capital E there.
Will produce oxygen and C6H12O6.
Okay, I'm going to go real general with carbohydrate right there. Solar energy. Okay, so the reactants are CO2 and water. And then Grant's right, you do have to have some solar energy. And you know what else? You got to have a particular pigment. This is on our lecture test Thursday. What's the name of that pigment you have to have? Chlorophyll. A, gotta have that too. There's no photosynthetic cell on Earth that doesn't use chlorophyll A. And then the products are oxygen and carbohydrate. Oxygen and carbohydrate. And this is on page 67. If you want to put a box around it or make yourself a note to look on page 67. Page 67. Second part of number 1. In what plant organelle does this process called photosynthesis occur? Chloroplast. And a picture of the chloroplast is also on page 67. I think it was on our lecture test last week too. Do y'all remember that? Me too. Okay, page 68 for number 2. So we did this procedure called paper chromatography. You may have taken some notes on page 68. What solvent is used in paper chromatography? And I'm going to give you 2 choices. Did we use a polar solvent like water, or did we use a non-polar solvent like ether? It was non-polar. And I want to remind you, we picked a non-polar solvent because the pigments are also non-polar. These pigments won't dissolve in water. Okay, how does paper chromatography work? Well, first of all, what's the goal of paper chromatography? What's it try to accomplish? Thank you. You already said it separates molecules. I think she's getting this off of page 68. And then it separates molecules based on something. Based on what?
Solubility.
Good. And again, you may have written this down on page 68. Now this is pretty cool, and I recall that you all did a really good job on this. You really mashed that leaf and got a nice pigment band, and then it separated really clearly. And on page 68, there's a picture that shows you the pigments that we got. Okay, so I'm going to go through those pigments and make sure that you know these. Now, remember our test is multiple choice. You don't have to memorize them specifically, but you do have to know them. What was the top pigment in our chromatogram? What's its name? Carotene. That's good. And it was yellow for our leaves. Carotene, yellow. Carotene's very common here in Texas. Okay, then there was a space and there was some more yellow. What's that second yellow? It's called xanthophyll. Xanthophyll, another carotenoid. So chemically, carotene and xanthophyll are very similar to each other in color and their chemical composition. Okay, then we hit this really gorgeous green. It was just gorgeous. Okay, what's the name of it? This one's chlorophyll A. And the last one? Chlorophyll B. Okay, then I put in this little question. If those were your choices right there, which one is the most nonpolar? The one that went the farthest, carotene. Carotene is our answer because it went the farthest, it dissolved the best in the solvent, and it went to the top. Now, guys, you're going to have to know these 4 pigments in order. Carotene's first, xanthophyll second, chlorophyll A, then chlorophyll B. Know them in order. I mean, I'll give you all the names, but know who was first, know who was second, who was third, who was fourth. Okay, you have to know carotene was the most nonpolar. Who was the least nonpolar? Chlorophyll B. Everybody okay on that? Chlorophyll B. Super. Okay, question number 3. Let us all go over to page 72. Now, as we're going, can you just stop for a second on page 70 and let me tell you that page 70 is a nice page because on page 70, we see that white light is actually made of all the colors of the rainbow. Just want to remind you about that. Great. And on your way over, look at page 71. You see that lamp shining through that beaker of water, and then the light would come through the water to the plant in the test tubes. Everybody see it? What gas would that plant be making? Oxygen. Because what process would that plant be doing? Photosynthesis. Well done. Now, page 72. Okay, on page 72 it says study the absorption spectrum on page 72. What colors of light does chlorophyll absorb the best? And let's go ahead and do chlorophyll A, because we know that's the most important one is chlorophyll A. So take a look at chlorophyll A. What colors of light does chlorophyll A absorb the best? Violet. Does everybody agree on violet? Okay, violet. Also known as purple. Anything else that you want to mention? Red is very nice also. Big peak in red. What color or colors of light does chlorophyll A absorb the least? Green. And on into the yellow. If you want to mention yellow, got a little pretty low in the yellow there. Okay, how did you know which ones it absorbed the best? What'd you look for?
I look at the picture.
Yes, but what'd you look for on the picture? The peaks. Wouldn't you call those peaks? Peaks. And what'd you look for to know what it didn't absorb? The low parts, the valleys. Okay. And the reason we did that is if you look at the y-axis is absorption. So if you see a peak, that means it's absorbing very well. And if it's not, then you'd have a valley or a low spot. You okay on reading that graph? Okay. Okay. Well, still on that same page, there's not just one pigment there. There's 3 different graphs superimposed there. We have the pigment chlorophyll A, the pigment chlorophyll B, and the carotenoids. And we found that the leaves that we mashed had all of those. That's very common for a leaf to have all of those. And so I have this question. Why is it beneficial to have several different plant pigments involved in photosynthesis?
They can absorb different kinds of light.
Did we have this question on the test last week? If plants could absorb every color of light, what color would the plant be? Yeah, what would the answer?
Black.
Black. Wouldn't that be weird, looking out the window, everything be black? Different.
We have healthy plants, though.
They'd be very healthy. Sounds like something from a science fiction show, black plants. That'd be cool. Maybe that could be the name of the movie. I digress. Okay. What color of light is best for photosynthesis? I hear violet and blue. Do you want to vote for violet and blue?
Yes.
Who told me that, Michaela?
Wait, yes.
Oh, yes. Now you change your vote. Why is Michaela right? Why is white the best color of light? Because it's all the colors. That's very good. What would be the worst color of light for photosynthesis? Green. Definitely green. Everybody ready for number 5? Okay, true or false? Most plants carry out photosynthesis only during the day. That's true. Because of the photo part of photosynthesis. What's the photo part mean? Light. You got to have light. Ian, you like that okay? Look very pleased with that true-false statement. I guess. True or false? Plants carry out cellular respiration only at night. I have a vote for false, and I agree with it. When do plants do cellular respiration? All the time. Plants respire all the time. Every living cell respires all the time. If you stop respiring, you die. So this is false because plants respire all the time. Every day, every minute, every second, they're respiring. But okay, on that part of the true-false question. Okay, now this particular question, please turn to page 74.
I like this.
Yes, I like this experiment too. And I drew it here. Okay. So first we took a beaker, put water in it. We put this pH indicator called phenol red into it. And then, do you remember how we put CO2 into the water? Blew with a straw. And we blew until the phenol red turned yellow. So just check your notes on page 74. If it was yellow, that meant that we were high in CO2. I think this is in your notes on page 74. If it was pink, oops, then that meant we were low in CO2. That's just something to bear in mind as we look at the next question. But when we blew CO2 into the water, the CO2 combines with water. What does it make?
Carbonic acid.
Carbonic acid, which is H2CO3. I was telling you guys that I was worried about that because of global warming, because we're putting so much CO2 into the air and it's going to combine with water out in the oceans. Kind of worried about that. But that's not the problem right now. The problem is you've got a test tomorrow. So let's stay focused. So yeah, we made carbonic acid, which lowered the pH. That's why the phenol red turned. It's almost magical every time it happens. I think it's cool. But anyway, we put one of the beakers in the light and one of them in the dark. I have, why did the solution turn back to red, also known as pink, after being placed in the light? So this would have been the beaker that we put in the light. If you'll check your notes on page 74, I think that we wrote down that when we came back, the beaker that had been in the light, it was pink. Is that true? And then why was it pink? Okay, so the plant did what? Plant did photosynthesis. I'm going to abbreviate that. And then how did the plant doing photosynthesis influence the amount of CO2? Did it raise it or lower it?
Lower.
Lowered the CO2 levels. That's right. So I'm just going to check. When I say there's low CO2, that means it's pink. Correct? Okay. Why did the solution stay yellow when placed in the dark? And so that's supposed to be my dark symbol for...
Because
Okay.
there's no solar energy.
Okay. So...
Not that much oxygen either.
Okay, well this has nothing to do with oxygen. You got no solar energy. What does that mean? No photosynthesis. Good. You can't do photosynthesis without some light. Okay, so the CO2, did the CO2 stay high? Yes. Matter of fact, it probably went up. How? How would it have gone up?
Cellular respiration.
Cellular respiration. That's right. You probably had more CO2 the next day than you did before because the plant was respiring. But okay, on that activity. Okay, question 7. Why is it important to have autotrophs, heterotrophs, and decomposers in an ecosystem? Well, guys, I drew an ecosystem. Let me tell you what's here. So it's like a little aquarium, and I have rocks on the bottom. These are little rocks right there. And I have a plant right here. And then I don't know if you can tell, but these are fish. I did not take art class, okay? Those are my fish. And these fish poop. Fish poop. I don't know if you know about that, but they do. Okay. So, oh, and I have sun. Now, I have that the plant is going to absorb energy from the sun and do photosynthesis. So check it out. The plant's going to make oxygen. It's going to make carbohydrate. Who's going to use the oxygen? The fish. And who's going to eat the carbohydrate? The fish. And when they do that, then they make carbon dioxide and they make water. What process are they doing? Cellular respiration. And then the plant uses the CO2 in the water to do photosynthesis. Does everybody see I got a cycle going on there? Okay, in this cycle, who are the autotrophs? The plants. Who are the heterotrophs? The fish. Who are the decomposers? Nope. They're living on the rocks. That's why you put the rocks down there. It's bacteria. Why do you need the bacteria living on the rocks? Break down the fish poop. Okay? Or like some of those leaves on this plant are going to die and fall to the bottom. You got to break down that dead leaf, right? So do you need the rocks at the bottom? Yeah, so the bacteria have a place to grow. So everybody understand that decomposers are super important. Okay, now, I don't know if you noticed, but I got a lid on this. Here's my lid. Would this ecosystem stay healthy longer with the lid or without the lid? I agree with Michaela. If I took the lid off, it's going to be better. Why? There'll be more gas exchange. More oxygen can come in, more CO2 can dissipate out. So having the lid off is going to live longer than having the lid on. Everybody okay with that idea? Just for example, I got my turtles back there. Everybody knows those turtles are right back behind Amanda. It does have a lid, but if you look at the lid, it's not a solid lid. What kind of lid is it? Yeah, it's a net. It's a screen. It's a screen so that the air can still go between the aquarium and the atmosphere. So it does gas exchange, but of course the lid's to keep those turtles in there. Okay, they could get bigger. They're growing really fast. I don't even know what's going to happen back there. Every time I look down, they're twice the size they used to be. They're just, yeah, you may see them in Armand Bio one day because they're just getting so big. They're cute. All right, is everybody okay on that question? Okay, so would you glance back at exercise 6, photosynthesis, and see if there's anything there that we may have missed or you need repeated? Okay. Page 77, please. We're going to cellular respiration. And guys, on page 77, I'm going to start with aerobic cellular respiration. That's the chemical reaction that's on page 77. It's C6H12O6 plus O2 makes CO2, water, and ATP. But you have to know how many ATP. 38. Thank you very much. 38. Okay, question, class. How did I know this one was aerobic? Because it's got oxygen as a reactant. So it's the O2 right there that makes this aerobic. Again, this is on page 77. And I think on page 77 we did put the 38 there in front of the ATP. Is that correct? Okay, you got to know that. You got to know what makes it aerobic, and that's the oxygen. Yep, there it is. Okay, then I have these two up here. Glance up at the screen real fast. These are not aerobic. How do you know?
No oxygen.
No oxygen. So you could classify those as anaerobic. These are anaerobic. Now, if you would like to find them, page 78. Let's find them on page 78. One of them is already written there on page 78. I think it's the top one, where glucose breaks down into carbon dioxide, alcohol, and 2 ATP. That's called alcohol fermentation. But we put the second one right there on page 78. Didn't you squeeze it in right underneath the sentence that says when animals, such as human, you wrote it in there? Okay, that's this second one. Probably has a 2 right there. Okay, I want to ask you about these two. Everybody agrees they're anaerobic. Okay, I want to ask you when you would use each one. Okay, when would you, or which one would you use? And we'll just call this letter A. Let's call this one letter B. So just refer to them as A and B. Which one would you use if you were trying to make bread? Why?
Yeast.
The yeast, and the yeast is making
Carbon
carbon dioxide. And what does that do to the bread?
Makes it rise.
Makes it rise. So we use letter A to make the bread rise. Shake your head. Okay. Which one of these would you use if you were trying to make sour cream? It'd be B. Because lactate is actually an acid, and that makes it sour. Yeah, that makes it sour.
This is a reaction done by bacteria.
Bacteria, that's right. Instead of yeast, it's bacteria. You're right. How about if you wanted to make beer? Which one would you use to make beer? A. Why?
Ethanol.
The ethanol, this part. You want this part right here, the alcohol. Right there. And again, you'd have to use yeast to do that. Right? Right. Okay, let me ask you this question. Which one is going on inside your muscle cells when you're working out too hard? It's B. Good. B. That's the one you can do in your muscle cells. And how do you know when you're doing letter B? The burning. Your muscles start to burn because of the acid. Everybody okay on these two chemical reactions that are on page 78? Alrighty. I'm going to take out the word both, and I'm going to change it to all, because there's actually 3 chemical reactions. Let's make sure we have all 3. Page 77, there's 1. And page 78, there's 2. So really, all 3 of them start with what molecule? Glucose. That's good. Okay, you get to pick from all 3 of them. Out of all 3 of them, which one do you think the glucose is completely broken down and all the energy is harvested? Aerobic. Okay, you are going to have to know this. How much ATP does each reaction yield? You have to know 38 for aerobic. And then what about the anaerobic ones?
2.
Just 2. Got to know that. Just 2. Okay, guys, I'm going to jump over to page 82. Page 82 for number 4 on cellular respiration. It says, be familiar with the fermentation experiment, i.e. solutions that we used, what we did. So I drew this up here. I have the test tube number 1. Just check your notes here on page 82. Was supposed to be glucose and yeast. Test tube 2 was fructose and yeast. Test tube 3 was sucrose and yeast. Test tube 4 should have been distilled water and yeast. Is that what you have in your notes? Okay. And then we measured a gas bubble in each one. And I think we wrote it down under net change. And I also believe we measured it in millimeters. Is that correct? Okay, before we get started, what was the identity of the gas bubble? Like, what gas was the gas bubble?
Carbon dioxide.
Carbon dioxide. We were measuring carbon dioxide. Carbon dioxide. You probably wrote that down on page 82. But we were measuring carbon dioxide. Okay, and then how much carbon dioxide there is an indication of how well the yeast cells could ferment the sugar. Now, just check our class data. I usually like class data over individual data. On your class data, which sugar had the best fermentation? Glucose. And so I have that here too. See, my CO2 is right there, and I have the most CO2 on number 1. I think number 2 came in second place, number 3 got third place, and number 4 was dead last. Now, we all understand why number 4 didn't ferment very well. How come number 4 did not ferment very well?
It was water.
Because it was water. Can you use water for energy? No. But you can use sugar for energy. Why did sucrose perform poorly? Because it's a disaccharide. What does the yeast have to do before it can use the sucrose? Break it down. So these first 2 are monosaccharides. Glucose and fructose, they're monosaccharides. So they're ready to roll. Cells could use them immediately for energy, whereas sucrose had to be broken down. So make sure that's in your notes there on page 82 about how sucrose has to be broken down before our cells can use it. Also make sure you know the gas bubble is CO2. We just did that. I'm just answering the questions. Did the gas bubble increase or decrease with each, and why? Well, the cells respire using the sugar, so anything with sugar in there, the cells will respire, so you get an increase in the gas bubble. And what was the gas? It was CO2. Okay, the aerobic cellular respiration experiment. That was my poor attempt to draw it. I already told you guys I didn't take art class. But in case you didn't remember it, I also set some up back here. I'm going to look at them again. They're the ones that look like this. This is the day we had the shaving cream. That's what makes it memorable. Shaving cream. Okay. So I was trying to draw it up there. You can see I have a ring stand with a clamp holding a test tube. I put cotton in the bottom, and you guys soak the cotton with KOH. Then you put some peas on top of it. And then we put a stopper in it that had a pipette coming out of it. We shot shaving cream into the end of the pipette. You guys remember that? Okay. So let's look at our data on that. And I believe our data for that activity is on page 80. Page 80. Now, you all surprised me so much that day because I was expecting test tube 1 to have the largest amount of movement of the shaving cream, but I remember that's not what happened. Which test tube did our class have the most movement on? Test tube 2. Yeah, test tube 2. Which did prove to us that even though it was full of peas that were dry, those peas were alive and they were respiring. I think it was, Michaela, your table that had, such a dramatic increase in the fermentation right there. It's pretty amazing. All right, what will I ask you about this one? I'm going to ask you what this activity was supposed to show, and your choices would be like photosynthesis, aerobic respiration, anaerobic respiration, you know, the pathways we've been studying. What would be the answer? What did this this activity show? Aerobic, because we left plenty of air in those test tubes. We used big test tubes and we left plenty of air in there. Plenty of air. Okay, now this next question is almost the same question again. You told me this was aerobic cellular respiration. What gas would the peas be using inside the tube? Oxygen. How come I'm asking the same question again? How is that? Because the word aerobic means with oxygen. Okay, so the peas use oxygen. Okay, now I know Janet can say and testify. Janet, isn't there aluminum foil around those? Now, I left a window of aluminum foil open on each tube so you could look in each tube to see what was inside. But you remember the day we actually did it, we didn't leave a window open. We completely covered each tube with aluminum foil, yes? Why? Prevent photosynthesis. We were trying to keep the peas from doing photosynthesis. So I would ask you, why did we wrap the tubes with foil? I would ask you that. To prevent photosynthesis. Okay, question 6. Which direction did you expect the shaving cream to move, in or out? In. Why?
Because the peas would be trying to bring in air. And the shaving cream was floating up the only available hole.
I'm going to use word sucking. I don't know why. It just sounds appropriate. We're trying to suck in the air. Yep. And then Grant said, but they couldn't because we blocked the only opening with shaving cream. You remember we talked about why I chose shaving cream? Because it's slick. Good memory. Because it's slick, so it'll move very easily. Good job. All right, everybody already knows the question, answer to question 7. What gas was taken up by the germinating peas? I'm ready. Oxygen, good. Okay, so glance back over exercise 7, please, and see if you need anything clarified or repeated. Blair?
On number 4, when you said cells respire using sugars,
Mm-hmm. Yes, ma'am?
was it increase or decrease?
So the bubble should increase because the cells respiring, they're going to use the sugar and they're going to produce CO2. So the gas bubble should increase. Good question, Blair. Anybody else? Oh boy, this is going to be just like what we already did today. Doesn't it look familiar? It does to me too. Okay, so first let me tell you that what we mostly need is going to start on page 86. Yeah. Okay, so let's just all look at our choices. It's kind of like doing clicker questions. Look at your choices, and I'm going to describe it. Your choices are what's on the screen. I'm going to describe it, and you pick. If you don't remember too much about mitosis, or you just like to have a nice reference page number besides your personal lecture notes, page 88 is good too. It describes each stage of mitosis on page 88 and 89. That's a valuable page 88 and 89. Okay, so I'm going to describe it. You tell me which one this is. This is where the chromatids are pulled to opposite poles of the cell. Anaphase. Okay, that's good. This one is where you see nuclear membranes reform, nucleoli reform, and chromosomes relax. It's good. It's telophase. How about this one is where DNA copies itself? Interphase. That's during interphase, the DNA copies itself. Okay, this is where the nuclear membranes disintegrate and the spindle fibers start to form. It's a prophase. How about chromosomes line up in the middle of the cell? See how that's going to be really easy verbally, right? Okay. All right. Okay, there's a picture of our cell cycle, and this one's actually the one from your textbook. But a nice reference page is page 86 in our lab manual. Guys, the only difference is this G0. Everything else is the same. So again, since we just chatted about this, I'm going to go ahead and move forward. There's the cell cycle. Okay. Okay, know the form of DNA in each stage of the cell cycle. Now, first let me point out that I have drawn DNA in 2 different forms up here. One of them's red, one of them's blue, and one of them is chromatin, and one of them is chromosomes. Now, which one I got the blue one over here. I got the red one over here. Which one's chromatin? The red one. Why?
It's uncurled.
It's uncurled. It's all spread out. Okay, and then the blue one is tightly coiled. So be able to tell chromatin from chromosomes. Everybody okay on that? Yes? Okay. The other words I have up here is when can you find them? Well, chromatin you find during interphase. Chromosomes you find during mitosis. And again, we did that in lecture today, but if you need to indicate these things in your notes, feel free to do so. And again, this one over here is chromatin. That one's chromosomes. Chromatin exists during interphase. Chromosomes exist during mitosis. Okay? So check out my cell cycle again. It's the same cell cycle I had before, but I drew chromatin and chromosomes on it. Go check it out. Start at G1. Everybody look at G1. I would say my chromatin is single-sided there. Everybody shake your head. Single-sided. And then look at S. Now I'm over here, guys. Now I'm at S. I would say I still have chromatin, but now it is double-sided. How did it do that? It replicated. Because it says that during S, DNA replicates. Okay? And then look at G2. It's still double-sided. It's still chromatin. It's double-sided. Now, what's the difference between what happened in G2 and what I see in prophase? What's the difference? It's way shorter. So now is it still chromatin? What is it now? Chromosome. See, it converted from chromatin to chromosome. Okay, it's double-sided chromosome in prophase, a double-sided chromosome in metaphase, but, oh, wait a minute, what is that? Is it still double-sided?
No.
Oh, why? What happened?
It got pulled apart.
It got pulled apart because it gets pulled apart in anaphase. So now it's single-sided again. It's single-sided there in telophase, and look how it's still single-sided in G1. Still single-sided. What's the difference between these two? They're both single-sided, but in telophase, how's that different than what's in G1?
It's cool.
Yeah, it's uncoiled. It's relaxed in G1. Now, I start anaphase. Everybody look at my stars. I start anaphase right there, and I start S. Why? Why did I start those? What's significant about them? That's right. It's when there's a change. It's when it goes from double-sided to single-sided in anaphase, and then single-sided back to double-sided in S. So I starred those. Everybody take a look at it and see if it makes sense to you, how I have that drawn. So what's going on with the DNA? It's kind of what's the idea here. What's going on with the DNA? Everybody okay with that? Super. Oh, this is going to be so easy. And if you don't know what I'm doing, I'm on page 86 at the bottom of the page. I need to draw a line to identify the chromatid. What do you want me to draw my line to?
The tips.
Yep, the tips. Now again, chromatids are these two identical sides of a chromosome. Okay, and then where's the centromere? The middle, where the two chromatids are held together. Okay, on parts of a chromosome. Super. Okay, I got a few more definitions here. Cytokinesis. Help me out. What is cytokinesis? Division. Amelia's right. Division of cytoplasm. What are spindle fibers? Like, what do they do?
They attach to the chromatids and pull them apart.
I don't know if I can put a divide there, but I did. They are made out of protein, in case you're curious. They're made out of a protein. Oh, what's a centriole? Like, first of all, who's got those? Animals, good Dane. Animals only.
And they seem to produce spindle fibers.
Yeah, we're not sure, but they appear. Very conflicting evidence there. I have they appear to produce spindle fibers, and they're only in animals. Only in animals. Fascinating things. They're mysterious. The mysterious organelles. Okay, where did I get this from? This was all part of number 2. Chromatids, centromeres, cytokinesis, spindle fibers, centrioles, chromatin, chromosomes, all the words we've been using. Identify parts of a chromosome. Everybody look at number 2 and see if you need any help with anything there on number 2.
There's a full list on page 87.
Full list on page 87, I think at the top of the page. Even more than you wanted to know. Thank you, Grant. Anybody need anything on number 2? All right. Number 3, differences between plant and animal mitosis. So I'm just going to make this into a table. Looking for differences. Anybody?
Cell plate.
Cell plate. Plants use the cell plate. What do animals use? Cleavage furrow. Okay, now animals have an organelle we talk a lot about during mitosis that plants don't have. They may make spindle fibers, maybe they don't. What is that organelle? Centrioles. That's about it, guys. That's about it. So, plants and animals do mitosis very similarly. But okay, on number 3. Oh, there's my expert drawings of these things. Which one is the cell plate?
The first line in the square.
Bless you. Why is cleavage furrowing a suitable mechanism for animal cytokinesis but not for plant?
Because they don't have a cell.
Can I say it again?
They don't have a cell.
Yeah, so animals don't have a cell wall, and the cell wall is rigid. It cannot furrow. And of course, plants have the cell wall, thus they can't do cleavage furrows. Okay. I'm going to test you over mitosis in 3 ways. In 2 ways I know that you're very familiar with. The first one is when we built pop beads with yellow beads and red beads. You guys remember doing that? And if you'd like to study what they may look like, the stages of mitosis using pop beads, page 88 and 89 in the middle of the page. That's what we used to guide us when we made our pop bead mitosis simulation. But then we also did slides. We did animal mitosis slide and a plant mitosis slide. So the top of page 88 and 89 is the animal. The bottom of page 88 and 89 is the plant. That's the slides. Okay, then lastly, these things have been out for several days, but I'm sorry I haven't drawn your attention to them. Does everybody see them over on what would be the east side of the lab over here? There's models of animal mitosis. Those are the blue and white ones over kind of towards Ian. And then over here by Michaela, those are the plant mitoses. It would be so easy to just glance at those blue models and know that's animal. How do you know? Because of the cleavage furrow. And then, Michaela, can you tell me, is there a cell plate there visible in those plant? Oh, yeah. So when you get a chance, maybe on the way out the door, take a look at those. If you'd like to take any pictures of them. The cards, the blue cards say the name of the stage for animal. The green cards say the name of the stage for the plant. So take a look at my models, the models that are over here on the side. Pictures, pictures, pictures. Okay, I think we're on the back. Okay, again, we just chatted upstairs about this, diploid, haploid, and homologous chromosomes. Okay, I'm going to give you multiple choice. You tell me which one it is. Everybody ready? There's your 3 choices up there on the screen. This word means one set of chromosomes. Okay. What kind of cells are usually haploid? Sex cells. Nicely done. Sex cells. Okay, this means that they're in a pair and they carry information for the same traits. Homologous. You're always in a pair. They show up in pairs, and they carry info for the same traits. Okay, well, you guys help me out with my last definition. The only thing left is diploid. How would you define that? 2 sets. Good. Remember, you get 1 set from your mother and 1 set from your father. So all of the body cells that make up you have those 2 sets of chromosomes. 1 from your mom, 1 from your dad. Okay, the pictures of the stages of meiosis are on page 96 and 97. Let's all go there together because I'll ask you some questions about that. Okay, we start out meiosis with 1 cell. Is that 1 cell haploid or diploid when you start meiosis?
Diploid.
It is diploid. How did you figure that out, Grant?
There's 2 sets.
2 sets. And everybody find the little box that says, I'm on prophase 1. Are you?
Yes, ma'am.
Okay. 2N. What's 2N mean? 2 sets. Is that haploid or diploid? Diploid. See, it even tells you you're diploid. Okay, everybody look at that beginning picture, prophase 1. What is going on there? Those chromosomes are all wrapped around each other.
Playing twister.
It looks like they're playing twister. But yet, in biology, we did not choose that word for it. We did not. Thank you. It's called synapsis. You maybe underlined or highlighted that word in the typed area underneath prophase 1. What's happening there is synapsis. They're wrapping all around each other. So everybody see synapsis going on? Okay. Go to metaphase 1. How did they line up in metaphase 1? Single file or with their partner? With their partner. Now, remember, they can line up where all the reds were on one side and all the blues were on the other, or they could have lined up like the artist drew. We call that independent assortment. Now, I'm a little confused. How come some of these red chromosomes have blue tips and some of the blue chromosomes have red tips? Crossing over. When did crossing over occur? Back in prophase 1. That's good. Okay, everybody take a look at anaphase 1. What separates in anaphase 1? Homologous chromosomes. That's good. Okay, go over to telophase 1 and end with the 2 cells at the end. Okay, the 2 cells at the end in this little time period we're calling interkinesis. Everybody focusing on those 2 cells. Are the cells that are right there at interkinesis, is that haploid or diploid?
Haploid. Sorry. Haploid.
They're haploid. And if you count the chromosomes, there's only 2 per cell. How many chromosomes were there when we started in prophase 1? 4. Now there's 2. Okay, check this out. Look at those 2 cells in interkinesis. Are they identical or are they different? They're different. So I want you to notice that first division accomplishes your goals, getting haploid cells and getting different cells. So I had a student ask me, if you get everything you want in the first division, why do you have to do the second division? Because you do have to do the second division. Why can't you just stop after the first division? What's the problem? Got too many chromosomes. Every chromosome is double-sided. Everybody see how every chromosome is double-sided? So the second division is simply to split the two sides apart. The second division, if you look at it, is just mitosis. You start with haploid cells, you end with haploid cells. No change in chromosome number. So the second division is just to get the chromosome number to the appropriate number and get all your chromosomes single-sided. Single-sided. Okay, so listen to my question, looking at page 96 and 97. Everybody ready? During what stage of meiosis does the chromosome number go from diploid to haploid? Anaphase who? 2? 1. And technically, we'd probably go with telophase 1 because that's where you actually divide the cells with their cleavage furrow. Hey, you guys, it's better in your books. This is so small. Find telophase 1 and find the actual photo of a cell right up above it. Can you look at that in your book? See the actual photo of the cell? Is that cell plant or animal? I heard both responses. Look carefully. It's definitely plant. Look, just because it's round doesn't mean it's not plant. How do you know that one's plant? Cell plate. It's making a cell plate. Does everybody see the cell plate? The artist drawing is definitely animal, but the actual photo is plant. It's actually a plant. Y'all see the cell plates? Just in case you're wondering what those are cells making pollen inside of flowers. Have you heard of pollen? Do you know what it is? It's dehydrated sperm. That's exactly right. It's male sex cells, dehydrated. What's dehydrated mean?
No water.
No water. Why would pollen want to be dehydrated? Flies better through the air. And then if you're dehydrated, you're dormant. Then if you land in the appropriate spot to hydrate, then you start growing. Where's the appropriate spot? Where does pollen want to land? No, it wants to land on the female sex parts of another flower. That's where it hydrates and starts growing. Anyway, that's what's going on in those actual photos is you're making pollen there. Pollen. But okay, on my question, when do you change from diploid to haploid? And the best answer is telophase 1. Okay, I'm going to move us over to page 101. This is really good artwork on page 101 because it shows what happens in a man's testes, that's at the top, and it shows what happens in a woman's ovaries, that's at the bottom. Okay, in a man's testes, the diploid cell goes through meiosis and makes 4 haploid sperm. That's what I was expecting. You? Page 101. Is everybody looking at the male picture at the top? You start with 1 diploid cell. It does the first division, you get 2. It does the second division, you get 4, and they mature into sperm. So listen to the question. Per original diploid cell in the testes, how many mature sperm can you get? 4 is the answer. No surprise, right? Well, women, we're a little bit different. Let's take a look at our egg production out the bottom. We're a little bit trickier. Okay, we start with our original diploid cell. It's called the primary oocyte, and it does the first division, and it does equally divide the chromosomes, but it does not equally divide the cytoplasm. And so you do end up with 2 cells after the first division. One is called the secondary oocyte. The other one's called a polar body. Everybody find the polar body. What happens to the polar body? It dies. Okay, ladies, that is what we ovulate right there. And the polar body usually gets ovulated with the egg. It all comes out together. Okay, then if you have intercourse during your ovulation period, then you increase your chances of a sperm penetrating the secondary oocytes. Everybody see the sperm coming in? Yes? If that secondary oocyte is fertilized, it will divide again and form another polar body. That way it gets rid of its extra chromosomes. Because right now, that secondary oocyte had double-sided chromosomes. It doesn't want to have all that. It's got to get rid of those extra chromosomes. So it does the second division after it's fertilized. I don't know why. I think that's risky business. Why don't we just do both divisions before we ovulate? I don't know why, but I do know all mammals do this. Every mammal does that. It's not just humans, it's every mammal. Your kitties do it, your puppies do it, raccoons do this, rats, mice. Everybody does this. It's a mammal. It's weird. It's weird. Anyway, eventually, you're going to end up with these little cells called polar bodies, and what happens to all the polar bodies? They die. So looking at the woman, which is oogenesis, looking at the woman, listen to my question. Per original diploid cell that starts oogenesis, how many functional eggs do you get? One. Okay, so sometimes students say, well, why is it that way? Why don't women make 4 eggs? Well, then you probably have 4 babies at a time. There's issue number 1. And your uterus is not designed for that. But even if it was designed for that, here's the problem. Everybody look at the size difference between an egg from a woman and sperm from the man. Everybody see the size difference? Who's bigger? Eggs. Why? Because eggs have lots of cytoplasm. Do sperm have lots of cytoplasm? No, sperm with lots of cytoplasm would be fat sperm. And would fat sperm swim fast? No, they'd never get there. All the skinny sperm would beat them. Okay? So literally, sperm show up with their chromosomes and that's it. Well, your baby needs more than just chromosomes. Your baby needs Golgi. Your baby needs endoplasmic reticulum. Your baby needs some dang mitochondria. Okay? Your baby needs some cytoplasm rich in protein, sugar, and lipids. Who's going to give your baby all of that? The egg. So that's why we make 1 big sex cell instead of 4 itty-bitty sex cells. You ladies also know our sex cells don't have to move very far. Out the ovary and down the tube. Sperm cells.
You got a lot of work.
Oh. You should see the length of just inside the male reproductive system, how far sperm have to swim. They have to come up from the testes into the abdominal cavity to get into the urethra, then come down the urethra and through the penis to be ejaculated into the vagina. Once there, they got to find the cervix, and it's only open when she's ovulating. Otherwise, it's shut.
They have to fight off things that are trying to kill them too. And
There's acid all in the vagina trying to kill the... It's true. Acid in the vagina trying to kill the sperm. Now, if the cervix is open, they have to swim through the cervix into the uterus. Then once they're in the uterus, oh my God, there's 2 tubes. And the egg's only in one of them, and they don't know which one, so half of them go the wrong way. Most of their buddies are dead by now anyway, so half of them go up the left, and the egg was on the right. I mean, yeah, it's tough. Yeah, it is tough. It's tough to be a sperm. So the chances of one of the sperm actually getting to the egg are very low. So what do men do to increase their chances of fertilizing the egg? Do they make 4 sperm only?
No.
They make millions of sperm per day. Millions. Because the chances of 1 sperm getting there are so low. So low. Glad we had this chat. Yeah.
Can we move on now?
Uh-huh. Very
Was
good.
there a documentary that covered all this on like the National Geographic or something like that?
You know, there may have been, and if you Google it and get me a good title, I'd love to watch that. I don't know that I saw that specifically.
It came out.
Like a nice mainstream. Yeah, it's pretty good. Okay. Oh, I talked for too long. Let me wake it back up. It just went into... Okay. So, on page 102, you and I did look at a slide of an ovary. And I told you, you got to know an ovary from a testis. So go back and review the ovary slide. You'll be able to spot the ovary. There will be not very many, but some of these big swollen pockets that have the egg inside. Those swollen pockets are called follicles, and the egg inside is the oocyte. And then the testis is on page 103. Now the testis is going to look like mini donuts. They kind of looks like donuts because the tubes of a testis are hollow. And when you look in the center of those donuts, you can see the tails of the sperm. It's pretty good. Those donuts, so to speak, are the seminiferous tubules. And if you cut a tubule, that's why it looks like that. You know, it's like cutting a tube. You're going to get something round. And then, guys, also, there's a pretty good view of the interstitial cells. Now, your interstitial cells are labeled on page 103 at the top. Everybody find it? But that's not where I would have labeled if I were doing it. Come to the bottom and find where it says 100 micrometers. Everybody see where it says 100 micrometers? Look right up above that to the left. There's a little slice of blue that kind of looks like a piece of pie, triangular. That's a big patch of interstitial cells. I have this question for you. Which part, and here's your two choices, seminiferous tubules or interstitial cells, which part makes sperm? Seminiferous tubules, that's who makes sperm. So what's the point of the interstitial cells? What do they make? The hormone called testosterone. Okay, the word hormone's not going to be on your test. The word testosterone specifically, okay? Okay, so our sex organs have two functions. Make our sex cells, make our sex hormones. Got that. Okay. We did that. I moved on because we already talked about synapses and crossing over. But I just want to remind you, crossing over, and if you want to back up with me a couple pages to take a quick look at crossing over, this was back on page 96 and 97. Crossing over is where the homologous chromosomes switch pieces with each other. What does it get you?
Different DNA combinations.
I don't like that. Okay, let me reiterate. You don't get new traits. You get new combinations of traits. Okay, so before I go to exercise 9, could you guys just glance at meiosis and see if there's anything there that you'd like for me to reiterate or talk about again?
Number 3.
Number 3. Okay.
Will we just be able to recognize them, or
So I'm going to literally just put the slide on the test, but cover up the label, and then ask you to name this organ. So you have to tell me if it's an ovary or a testis.
Oh, okay. So we won't have to identify specific parts of it.
Probably not.
Okay, because that's what it sounds like on the.
I think the specific parts help you know it's an ovary.
Okay.
Anybody else? Okay, I'm going to go to Mendelian genetics. And class, those of you, well, let me start over. Tomorrow at 7:30, Mendelian genetics will be our lecture topic. So it would behoove you to be there because everything I want to talk about tomorrow will be a review for the lab test tomorrow too. So it'd be great for you to be there.
So we're a little ahead in our lab.
We're a little ahead on our lab. That is true. Okay, so I'm going to start with some definitions. And everybody knows Mendel. We talked about him last week. Father of genetics. Okay. He's a good guy. So allele, I'm going to just go right ahead. Allele is a trait choice. So blue eyes would be an allele, brown eyes would be an allele, green eyes would be an allele. They're choices for a trait. Okay, so like last time we counted corn, purple kernels of corn versus yellow kernels of corn, those are alleles. Those are choices for the trait. Choices for the trait. Trait choices are alleles. Okay, genotype is actual gene content. Now, when we do genotypes, we use letters. The capital letter stands for the dominant allele, and the lowercase letter stands for the recessive allele. We use letters. Genotypes use letters. Use letters. Phenotype is what it looks like. So, yellow is a phenotype. Purple is a phenotype. Tall is a phenotype. Dwarf, that's a phenotype. What does it look like? And so you use words for phenotype. Words. Use words for phenotypes. It's easier to know a person's phenotype than their genotype. But yeah, that's got to be true. It's easy to see the traits. It's harder to know whether they're just dominant or if they have the dominant and the recessive. It's hard to know. Okay, heterozygous, hetero. Does hetero mean the same or does hetero mean different? Different. If you're heterozygous, you have two different alleles, like this. Big T, little t. Or big A, little a. Or big Z, little z. I don't care. Whatever letter of the alphabet you like, that's heterozygous. You have the dominant allele and you have the recessive allele. What would be your phenotype? What would you look like, dominant or recessive? Dominant. So see, that's where it gets hard to know an individual's genotype from just their appearance. Okay, what does homo mean? Two same alleles, yeah.
So would that be the only way to for the recessive gene to show?
Very good. If you're recessive, you're homozygous. That's very good. So recessive individuals, that's going to be the basis of our next mathematical calculation for lab test 3, is that if you're recessive, we know your genotype. If you're recessive, we know your genotype. Okay, I've got little t would be homozygous. What else could be an example of homozygous? Big T, big T. You okay on those words? Super. Okay, what's the expected phenotypic ratio of a monohybrid cross? Well, I'm just going to use the letter A. This is a monohybrid cross. And I want the phenotypic ratio, what they look like, what you expect them, how many you expect to be dominant, how many you expect to be recessive. So I'm going to draw myself a square. What's the name of the square? Punnett. Big A, little a. I had to squeeze it in because my screen's small. But let me see. Here's a dominant, here's a dominant, here's a dominant, here's a recessive. So phenotypic ratio. How many do you, or what percent do you expect to be dominant? 75%. And recessive? 25. Now guys, that is good. That is good for percentages. What if you were just doing ratios? 3 to 1. Everybody agree 75% to 25% is the same as 3 to 1? Yes? Okay. Monohybrid. So you have to know what that means. Monohybrid means big letter crossed with big letter, little letter. And you expect a 3 to 1 ratio every time. Doesn't matter what the letter is. You could use the letter P and it would still be the same. Okay. If you did a monohybrid cross of corn, would you expect the observed results to be closer to the expected ratio if you did 5 crosses or 50 crosses? 50 crosses. Because it has a larger data size or larger sample size. We try to get large sample sizes to get rid of randomness. It's the whole concept of getting large sample sizes for research purposes. Tomorrow I'm going to show you some of Mendel's data. He had huge sample sizes, like thousands of pea plants. And that's how he got rid of randomness. Okay, what's the expected phenotypic ratio of a dihybrid cross? Okay, that would be 2 traits. How come it's 2 traits? What's the part that tells you it's 2 traits? Di. And then they're both heterozygous. What's the part that tells you it's heterozygous? I'd use the di part. Sorry?
2 different allele types.
Yep. The word hybrid. What's a hybrid? It's a cross between 2 different things. Okay, so different. Notice A, big A, and little a. B, big B, and little b. Okay, now guys, we did this cross. I'm not going to draw it for you. I want you to check out your notes from last Thursday. We called these handwritten notes genetics. Let's see if you can dig through your lab notes from last Thursday and find it. Find it. I think we did pennies and nickels. So maybe you got something about pennies heads, pennies tails, nickels heads, nickels tails. Do you guys have it?
Yes, ma'am.
And we did a big cross. It's a nice size cross. And I'm looking for the phenotypic ratio, what it looks like. I think it was 9 out of 16 came out double-headed. And 3 out of 16 came out with head of one, tail of another. And 3 out of 16 came out in the other combination of head and one and tail of another. And 1 out of 16 came out double-tailed. Is that true? So we call this the 9-3-3-1 ratio. 9 out of 16, 3 out of 16, 3 out of 16, 1 out of 16. This should be in your notes. We did it with pennies and nickels. Who's okay on that? Dihybrid. Okay. The law of independent assortment. Okay, well, let me tell you about the law of independent assortment. I don't want to write it, so I just want to tell you where it's at. Page 112. So you don't want to write it either. It's in a blue box that says the law of independent assortment. Did you find it? Members of one allelic pair segregate or assort independently of members of another allelic pair. Therefore, all possible combinations of alleles can occur in gametes. There it is on page 112. Okay, let me show you how you used this the other day. The other day, we did big N, big P, little p with our pennies and nickels. And I asked you, what combinations of P's and N's could come from this individual. And here's what I think you told me. You told me a big N could go with a big P. You know what you said? Then you said a little n could go with a big P. Then you said a big N could go with a little p. And there's one more combination. What else could happen?
Little n, little p.
Little n, little p. Do you remember doing that? That's the law of independent assortment, that every possible combination is equal in probability. Whenever you were putting these combinations of p's and n's together, you were doing the law of independent assortment. So who cares? Well, what does it get you, this law of independent assortment? Does it get you anything? It gets you more variation. Do you see what I'm talking about? Let's say that if the law of independent assortment didn't work, and that big N always went with big P, and little n always went with little p, and nothing else could happen, would you get as much variation? No, because you wouldn't have as many combinations. You wouldn't have as many combinations. But because of the law of independent assortment, we know that that's not what happens. Every combination is possible. Every combination is possible. Okay, so then we worked on this math thing called chi-square. Everybody, page 119, please. And let's grab our calculators, too. And if you don't have one, you can come up and borrow one from my calculator box up here. The purpose of chi-square. You need a calculator? My calculators don't have square buttons, but you guys know how to square stuff, right? What does that mean to square?
Times by itself.
Times by itself. See, I knew you knew how. Okay, just look at the chi-square value table at the top of page 19. Its purpose is crystal clear just by looking at the table. What's the purpose of chi-square?
Determine if your hypothesis.
Or not. We had a whole lot of or not the other day. Dane and I did not like it. We like our hypothesis to be supported. Okay. Let's say you run the chi-square test and you end up with a probability between 0.8 and 0.5. Everybody look at 0.8 and 0.5. Your chi-square value came up between 0.8 and 0.5. Does that mean your data support the hypothesis or does not?
Yeah.
Yes, it does. That's a pretty good chi-square right there. So it's happy, yes. You're happy. How about this question? A probability value greater than what is considered acceptable? Like, what's the cutoff on not acceptable?
Okay.
Yes. It's got to be greater than 10%. And that's why we drew our line right there at 10%. In your book, I'm sorry, on your table, didn't you draw your line right there between 10 and 20%? Remind me your name, young man? Jake. Do you have your line drawn on your table? Michaela, check his. Okay. Okay. I don't really like number 7. I'm going to omit it. I like number 8. Let's go to number 8. And it's time to work some chi-square. It's time to work some chi-square. Okay, first of all, these are plants. So I'm just going to read it off of number 8 because I can tell I didn't write it all here on the screen. Suppose you counted 125 green tobacco seedlings. You know tobacco's plant, right? Just checking. Okay, suppose you counted 125 green tobacco seedlings and 29 white tobacco seedlings on several plates. Okay, have you guys ever seen a white plant? They exist. They're really cool. They're just plain white. White stem, white leaves. Yeah, they're cool looking.
How do they live?
Let's talk about that. First, going to do the chi-square, then I want to talk about those albino plants. That's good. Suppose you counted them. Okay, according to the chi-square test, do your results support the hypothesis that both of their parents were heterozygous? Okay, first I'm going to make us a key, and I think you'll agree with me, that green for plants is dominant to being white. Does everybody agree with me? Because big G would mean you can make chlorophyll, and little g means you can't. So the ability to make chlorophyll would dominate over the non-ability to make chlorophyll. Because that's how you get albino plants. They can't make chlorophyll.
Oh,
Okay?
sorry, never mind. I'll say it afterwards.
Okay, now using this key, help me write the parents. We're hypothesizing both parents are heterozygous. So using the letter G, how would you write that?
Big G.
Big G.
Okay.
So they're both the same? That's a monohybrid cross, correct? So we're expecting 75% to be green and 25% to be white. Because that's a monohybrid cross. But okay on where I got my expected. I'm expecting 75% and 25%. 3-to-1 ratio. But okay so far? Okay, I got to go to a fresh screen. So, to get this started, take this off. I'm going to take how many I had. Let me see. 125 green and 29 white. I'm getting this off of number 8. Let's add those up to see how many total we had. 154. Now these are actual numbers. So these are what we actually observed. We observed 125 green. We observed 29 white. Is everybody okay so far? All right. Now, our expected numbers are in percents. We've got to get them into actual numbers. So here's what I think we should do. I think we should, on our calculator, I think we should take 154 and multiply it by 0.75. I got 115.5. That's what I expected to get for green. How come I used 154? That's the total. Why did I multiply it by 0.75?
That's the percentage.
Percentage. 0.75 is percent. So I was expecting 115.5 plants to be green. Let's see if you can figure out how many were you expecting to be white. Everybody do it on their calculator. James said 38.5. You okay on how we got those numbers? All right, great. Then we're ready to do our chi-square. On chi-square, it is the observed minus the expected squared over the expected. That was for green. I'm going to do it again for white, if I can squeeze it in. You guys get your calculators out, and remember we're going to go 4 places behind the decimal. So start working on with your calculator. Let's see what we get. Keep your fingers crossed. Do you get something around 3.15? Yeah? Man. Okay, back on the table. How many degrees of freedom do we have? 1. Dang. Just missed it. Just missed it.
The worst luck with this.
We really do. Maybe we need a bigger sample size.
Where did you get the 0.8061?
Yeah,
You didn't get that? Okay, so maybe that's why we didn't get it.
it still doesn't make it.
Aw, it still doesn't make it?
7814. It makes it to 3.125.
You're right. This one is 7814. Okay. Thank you, Megan. So that changes it to 3 point what? 3 point 1256. Still didn't make it though, huh? Probability range still come out the same? 10 to 5? Well, poo. Okay, let's talk about albino plants. This is an excellent question. So to be albino, or a white plant, write this down. To be a white plant, you have to be little g. And James said he's seen them before. Who else has seen these white plants? Anybody else? Dane, you've seen them too? I've seen them because I go to Nebraska every summer, and you'll see them sometimes out in the cornfield. These albino corn plants. But they're only when they're young. Only when they're young. Okay, so how do the farmers, how come they keep showing up? So who would have to be the parents to get you this?
Big G, little g. Big G, little g.
Everybody agree that's how they keep showing up? Okay, this is correct. How come this wouldn't be it? Look at this cross.
Because then there would be more white plants than there would be.
It'd be a 50-50. But how come this would never happen? How come this cross is not even possible?
Do the little plants not survive or don't?
They don't survive. That's good. Okay, so these plants don't live very long. How come they're alive when James or Dane and I saw them? It's because they were young. What were they living off of? The food in the sea. The food in the sea. How come they're not going to live very long? They can't photosynthesize. So as soon as the food in the seed is gone, they're dead. They have no hope whatsoever. Poor little plants. No hope whatsoever. All right, so tomorrow is lab test 2. And I want to give you one more hint before you take off. On the lab test tomorrow, we're going to do this in lecture tomorrow too. You should be able to do these following crosses. You should be able to do this cross. This cross, this cross, and this cross. Those are the 4 crosses that you're responsible for both lab and lecture. Now I know we've done this cross and we have done this cross in our notes. So you might want to take a little bit of time tonight to work on those other 2 crosses. And then I'll field questions about that at the beginning of class tomorrow if you have questions about these other 2 crosses. But okay, on what crosses they're responsible for. Super. So I'll see you in the morning.